And Arthur's going to start his presentation now.
I'm going to talk about vaccines. It's a subject very much in everybody's mind these days. It's also a little bit of a complicated subject, but it's fascinating. Now, in order to understand some of these terms, you need Molecular Biology 101. So I'm going to simplify it as much as I can. You know, basically, human beings have evolved from microbes to man in 3.77 billion years, resulting in the most sophisticated organism on the earth today. We will glimpse into one facet of this organism, immunity. And this immunity is the one that leads to vaccine development. Basically, when you look at the egg, the fertilized egg is a single cell. And in this single cell, you have a nucleus. And inside this nucleus is a 2-meter long DNA coiled in a double helix, compacted into 26 chromosomes and packed inside. And it has 25,000 genes. And these genes are all marked by bases, which will have a base as AT, ATGC. These are the 4 bases. And they're all grouped into triplets, 3 bases together. Each base with 3 triplets will sort of code for 1 amino acid. Now we have 21 amino acids that we use in our system. These amino acids are the ones that form proteins. And proteins are the building blocks of our body. We have the structural proteins like the skin, the muscle, and all the various organs. These are all structures made of protein. We also have functional proteins. These are enzymes, basically. These enzymes are biocatalysts. These are catalysts that are used for reactions to synthesize new compounds. Like we synthesize all the hormones, the neurotransmitters, the bones, the teeth, the hair, the nails, and these are all synthesized by the proteins. What you need is, protein is the basic building block, and it is from this you get the rest of the human thing. And when you look at it, this single cell has the entire message for differentiating into a human being compacted into this nucleus. It's just simply mind-boggling to see how this evolution has taken place over so many billion years. Progressively, you see this fertilized egg differentiating. It's like a flower opening, petal by petal, and you have new things coming off, ultimately ending up in a complete human being. Now, we have to remember that DNA is the one that basically codes for all our characteristics. And this DNA is in the nucleus. It's in the nucleus, and we have a DNA. And in order for this DNA to start forming new proteins, first it is transcribed into a message. And this message is called a messenger RNA. And this messenger RNA migrates and comes out of the nucleus into the cytoplasm. And here there is a factory called the ribosomal factory. And this, in the ribosomal factory, the messenger RNA goes in and is translated and comes out as a protein. That's how proteins are formed. Now, all the proteins follow the same thing from the basic microorganism to the humans. This process is very common. So we have this transcription leading to RNA and translation leading to the protein of the 21 amino acids that are being formed. Now, you have this code, ATGC for the DNA, and in RNA, one particular base, T, is replaced by U. It becomes AUGC. Now, this will become relevant when we look at the viral RNA, the COVID virus. And there you will have only U because it's an RNA virus. Immunity is something that is basic, and it's a field, it is extremely advanced, mainly because it has so much impact on cancer treatment. We have so many different types of ways of attacking the cancer cells using our immune system. Basically, our immune system consists of two groups, innate immunity and adaptive immunity. The innate immunity is the first line of defense. This first line of defense, you have the white blood cells consisting of macrophages, neutrophils, dendritic cells, eosinophils, and basophils. As soon as a foreign organism comes in, these things attack and destroy this invading organism. We also have another system called the adaptive immunity. This one kicks in after the invader comes in. Once the invader comes in, it induces certain other cells called the B cells and the T cells. And these B cells and T cells are produced in the bone marrow and spleen, and then they mature in the thymus, which is a gland in the middle of the chest. And then they circulate in the lymphatic system and the blood vascular system. Now, these immune cells, the B cells, are the ones are of great importance because they produce antibodies. What these antibodies do is they go and mark the foreign invading organism. They mark it. I always remember the biblical story of how when the Pharaoh declared that every new firstborn should be killed, and the Israelites, they had to have the lamb, the blood of lamb on their doorsteps so that the angel of death will miss them. That's exactly what happens. Here, the antibodies mark these invading cells for destruction. Now, how is it that our own cells are not destroyed? Because as our cells are formed, there are a group of genes that produce markers. And these markers are marked on all the cells in the human body. And this is unique to each person, not the race as a whole, but each individual. Mine will be different from Peter's, or Peter's will be different from Saifu's. And we will have a unique system. And in our system, our own immune system will not touch these cells. But an invading cell that is marked by these antibodies will be destroyed. It can be destroyed by the macrophage. It's also may be destroyed by natural killer T cells and also various T cells. This is how they destroy invading stuff. Now, there are antibodies that are generally formed, and we use this methodology to study antibodies. Normally, the old-fashioned way of doing it is to inject an antigen. An antigen is the one that induces antibodies. Say, for example, when a bacterium or a virus invades a person, that is an antigen that will create antibodies against a specific virus or bacterium. If we want to sort of study a particular protein that we want to see whether to identify the protein, we take that protein, or in the lab, in our own lab, in the forestry lab, we used to identify viruses. So we would inject a particular, spruce budworm or some insect virus into the rabbit. And the rabbit, this virus will be the antigen, and it'll induce B cells, and the B cells will produce antibodies. And then we collect the serum, and we have the antibodies. Now, if you want to check whether we have the particular virus identifying, we add this serum to the virus, and then it sort of precipitates, and we can have a particular, reaction with some coloring material, some kind of a dye, and we can identify whether it is our virus or it is something else. If it is something else, it will not react. If it is the virus that we had as an antigen, you will get a positive reaction. Now, this type of thing, it produces memory cells, the beta cells produce antibodies of various kinds. Now, if you look at the antigen, it may be a single protein, but it will have different areas for which the antibodies can be produced. These areas are called epitopes, and these epitopes are on the surface of the protein. And these antibodies, each one will attack one epitope. So what you have is a collection of antibodies called polyclonal antibodies, where several epitopes of the antigen are recognized. More recently, we have a thing called monoclonal antibody. Now, if you remember, President Trump was treated by monoclonal antibodies against the coronavirus. Now, what you do for that is you have a mouse and you inject the antigen, and it produces the antibodies. But these antibodies are polyclonal. There's for all the various types of surface proteins. So what we do is we collect the spleens where the antibodies are produced, and we macerate it and separate the cells and add a cancer tissue line, cancer cell, myeloma cell. And they both fuse, and each individual fuses, and it forms what they call hybridoma cells. These hybridomas can be separated, and each hybridoma cell recognizes one epitope. So you have separated everything, and you can put them all in separate culture media and then let them all multiply. Each one in a little microcell will produce antibody against that particular isotope. So we can characterize that, and we can identify on which epitope it recognizes, and we have a monoclonal for each type of an epitope. Now, they produced one for the spike protein of the coronavirus, and they produced this monoclonal antibody. They were using this against the person. They were injecting this antibody into President Trump to control his viral infection. Now, induction of antibodies using antigens is the basis of vaccine production. Vaccine production has a history dating all the way back to 1796. And Edward Jenner coined the term vaccine, and he developed the first vaccine against smallpox. Since then, we have vaccines against rabies, tetanus, diphtheria, scarlet fever, and for tetanus, they have used a sort of a tetanus toxoid. Pertussis or whooping cough. Polio, they have the one that is injected, that's a Salk vaccine, and the oral one is a Sabin vaccine. Then against measles, mumps, pneumonia, meningitis, Lyme disease, Ebola, and now we have vaccine against COVID-19. So there are lots more of these things produced. Now, the COVID-19 virus is a very small virus. It's 100 nanometers in diameter, and it has got the genome inside. This is the genome. And this genome is a single long RNA chain. This RNA chain, when they detected it in China in December 2019, when the infection started. Then they sequenced this in China on January 11th, 2020. And when they sequenced it, they published it so that the whole world can use this sequence to study that. So whatever we say about China, they were quite responsible. When they got the thing, they spread this information around the world so everybody could work on it. The mistake was we didn't take it seriously and started working on it right away. Now, you know that this coronavirus has the spike protein. That's the spike protein it uses to enter the human cell. The host cell of the human beings have a receptor called the angiotensin converting enzyme receptor, ACE2. And this particular spike protein goes and attaches itself to this receptor, enabling it to go inside. And once it goes inside, it has got a single strand of RNA, which is something like the messenger RNA. And it uses the ribosome, the factory of the host cell, to manufacture more of these. And it is translated into viral proteins, and it takes over the machinery of the cell and produces more and more of these viruses. And of course, they attack the air sacs, the lining, the bronchial epithelial cells, and it destroys the lung. ACE2 receptor is primarily present in the cells of the lining of the nasal passage, lungs, and everything, and that's where you have the main line of attack. But it is wrong to think that it is only there. It has been shown that ACE receptors are present in various organs, in blood capillaries, in the nervous tissue, in the stomach lining. So you can have this virus invading all these areas. That is why you have these secondary effects all over the show. You know, not only the lungs and the nasal passage, but all over. Classical vaccines take 5 to 10 years to produce. And you have a preclinical stage, which takes about 30 months. And then you have phase 1, we have 7 volunteers, phase 2, phase 3, and approval of manufacture. Now, more recently, because we have a tremendous amount of background information, this whole thing has been truncated. Now, we can develop a vaccine within 1 to 2 years. Now, we have created this COVID-19 vaccine in record time. It's because of all the background information that we have. So the preclinical stage is no time at all. Phase 1 is 6 months. Phase 2 is another 6 months. Phase 3, there are already thousands of volunteers used. It doesn't take any time at all. And approval, they have in no time, 6 months. So within 1 to 2 years, you can manufacture a vaccine. That is why we have so many vaccines. There are, right now, there are about 200 vaccines that are being made all around the world. Only some have been approved. So I'm not going to talk about all the vaccines. I'm going to take about a few and tell them how they work. Basically, vaccines, you know, they come under different platforms. The first platform, like the tuberculosis, oral polio vaccine, measles, rotavirus, yellow fever, they use what they call live attenuated virus. In other words, the virus is treated with something like formaldehyde or things like that, or radiation, and then inactivate the virus. And they take the inactivated virus and inject it into, an animal, and produce antibodies. That's a classical way of using antibodies here for these vaccines. Then you can have killed antigen. You have the antigen is completely destroyed and use the killed antigen. This thing is not really killed, but sort of they are inactivated. Then you have subunit. You take a part of the antigen and use that. Then you have some of the toxins that are produced by the virus and use that, like tetanus, where the toxide is used. More recently, we have an RNA-based system where you have the RNA of the virus and use that as the antigen and produce antibodies. Now, all these antibodies are mixed with adjuvants. They have to have various carrying agents and things like that. And that is why you have to do all the safety tests, because all these carriers have various types of side effects. So you try and check the safety of all these various adjuvants. But since we have been using these adjuvants in the production of various vaccines for so many years, we know which ones are toxic and which ones are not. So we have a tremendous amount of background information, and we can very quickly produce these adjuvant mixture that are quite safe. Now, these are the common types, the first four types that are being used. And the second is using attenuated viral vectors. Now here what they do is, you take a virus and then you engineer the virus so that it doesn't work as a disease-causing virus, but it's just as a virus that enters a cell. We call this a viral vector. Now, we've been using these viral vectors in molecular biology for studying various types of gene introduction into cells. So, using these viral vectors, which are actually inactivated vectors that have been thoroughly studied and we know the structure, these are all circular vectors, and in which you can introduce the antigen, and this is delivered as this vector. AstraZeneca vaccine is one example. I'll go into details later on. RNA vaccines are a brand new technology where the messenger RNA coding for spike protein is delivered into the cell where it is translated into the antigen protein. The Pfizer-BioNTech vaccine and Moderna are the ones that they use this mRNA method. Johnson & Johnson uses DNA instead of RNA to introduce into the cell. There are other types but are not ready for use. Example, in Quebec, they are working on a vaccine that is being manufactured in tobacco plants. The AstraZeneca vaccine uses a vector. This is in the Oxford vector. They use a chimpanzee adenovirus vector. This is an inactivated vector. All the disease-producing part has been removed, and the only thing it has, it carries itself into the cell. That is why you call it a vector. They put the spike protein DNA into this insert. Now, you know that the RNA virus, the COVID-19 is an RNA virus. So in order to take this RNA virus, they have to use a special enzyme called reverse transcriptase to change this RNA into DNA. And then once they change it into this DNA, you get the DNA of the spike protein. And they change this, and they use this, and they insert it into this chimpanzee adenovirus vector. And this one uses a special receptor called CAR receptor on the surface of the human cell and enters the human cell. And it forms a bubble with the spike DNA. And this bubble breaks, and the DNA is injected into the nucleus, the spike DNA. And the spike DNA nucleus, when it's inserted, it is converted into messenger RNA and sent outside into the cytoplasm. Once it goes into the cytoplasm, the mRNA is translated in the ribosomal factory into the protein for the virus. Now here we have the B cell. Once it comes out, it induces the reaction producing antibody. The antibody is a B cell formation. And these B cells immediately use this antigen and they produce antibodies against this spike protein. And then this B cell locks into the spike protein upon activation by helper cell produces antibodies. So you have a tremendous number of these antibodies, and these antibodies attack the spike protein and neutralize the virus. That's how the AstraZeneca vaccine operates. The Pfizer-BioNTech, on the other hand, they take the messenger RNA itself, and this messenger RNA, this sequence is there. They modify the sequence. I told you they have this sequence of the 4 bases, A, U, G, C. And they took this U base, which is uridine, and they changed it to a pseudo-uridine to stabilize the mRNA. Now, many of the things that they are using are all proprietary information, so I couldn't get my hands on all the details, but this is the general outline. Now, once I have this sort of, the mRNA, which is highly labile, is sort of stabilized by this modified change. And then they pack it in a lipid coating. The reason you pack it in a lipid coating is not only to protect the messenger RNA, but it enables this lipid-packed virus to enter the cell. So once I have this little lipid package carrying this modified mRNA sequence of the spike protein, it enters the cell. Once it enters the cell, it produces, all the proteins for the spike protein, They come out and they produce antibodies, and these antibodies neutralize the viral protein. Now, there are several vaccines that are right now being utilized. The Oxford AstraZeneca vaccine uses the viral vector, the chimpanzee vector, and you need 2 doses. It is 62 to 90% effective, and you can put it in a regular fridge, plus 3 to 5 degrees centigrade. The Moderna vaccine is a part of the virus. It's very much like the Pfizer-BioNTech. It's an RNA vaccine. You need 2 doses, 95% effective. You can keep it in the regular freezer, minus 20 degrees. The Pfizer-BioNTech is more sensitive. It needs 2 doses, 95%, but you have to keep it under dry ice, minus 70 degrees centigrade. The Russian virus, Gamaleya Sputnik, they use a human adenovirus vector. And this is a very special vector that they developed. And in this vector, they introduce the viral genome, and this viral genome produces the antibodies. You need 2 doses, 92% effective. You can keep it in a regular fridge. Johnson & Johnson also uses a virus vector, which is also a modified human adenovirus, and they use a DNA part, but they have only one. You have to admit only 1 dose, and it's 90% effective. You can store it in the regular fridge. But also cost-wise, the Oxford-Zeneca is the cheapest. Next is the Johnson & Johnson. The RNA viruses are a little more expensive. Now we come to this thorny problem of mutation. We have heard about how there are mutant varieties of the RNA virus. Originally, we thought there will not be any mutation because the spike protein is so specific and it needs the spike protein to enter the human cell that if you have antibody against the spike protein, you'll be very well covered. Unfortunately, that's not true. Now, in order to understand this mutation, you'll have to see how every gene that codes for a protein has a sequence of all these bases. And all these bases are arranged in triplets, and each triplet codes for a particular amino acid. So if you change one code, for example, from AAU to AAA, it'll become lysine. So in other words, the protein that it produces will be different because an amino acid will be changed. This is what is called a point mutation. This is the most common mutation. There are all kinds of other mutations. There are some mutations that are harmless. There are some mutations that are destructive to the virus. But these point mutations are the ones that we are encountering right now. Now, if you take the spike protein itself, it consists of three proteins forming a trimer. And in this trimer, right at the tip, there is a region called the receptor binding region, or receptor binding domain, RBD. This is the one that sticks to the ACE2 receptor of the human being to enter inside. And in this receptor binding domain, if there is a change in one of these amino acids, then you'll have a modified, it's a mutant. Now, to produce the mutants, a virus has to multiply many generations. You know that the exponential rate of replication greatly increases the possibility for mutation. Just one moment, please. So the more rapidly an organism multiplies, the more the chances of mutation. The virus has a replication rate that is really explosive. So the rate of multiplication is so great that the chances of mutation is also equally great. So you have to expect these type of changes. Currently, there are 3 types of variants that are known. the UK variant. Now, if you look at this thing, you have the spike protein from the coronavirus. This is the genome of the coronavirus. The spike protein, it's a trimer, 3 proteins. And then this is the receptor binding domain. It attaches to the ACE2 receptor and gets into the human cell. Now, all the variants, they are happening at the RBD, or receptor binding domain. The UK variant has one amino acid change. It's asparagine to tyrosine. Now, you know this virus genome, it's just a long bead of bases. And you can count the number of bases and you number them 1, 2, 3, 4 and all the time. And you have 29,903 bases. That's what they have figured out. And in this 29,903, you can take the number. What they found in the UK variant, its change is 501. The number 501, there is a change from asparagine to tyrosine. This is a one-letter code for amino acids. They say N to Y. So that's the change that you see. This is what you will see. And what this does is it increases the binding affinity. And also it creates a new point of contact. As a result, this UK variant is more infectious and it increases virus replication. That is why it is better than the wild type that we have been having. This one will be increasingly infectious and have more virus production. The South African variant, you have the same number as I told you, from 1 to 29,903. There is a change in 484, and there is another change in, I can't see this because you guys are staying here, and I'll move you back. The other change is 417. So you have this 484 and 417. So 417 is a glutamic acid to lysine. 417 is lysine to asparagine. This E484K is the one that is very worrisome. This is the one that makes this virus more deadly, because the AstraZeneca virus does not work on the South African variant, mainly because of this mutation. It becomes more infectious and less susceptible to vaccines. The Brazilian variant has 3 mutants. There is one at 484, one at 501, and one at 417. Now, if you see, the 501 is the UK variant. It has got that. It has got the South African variant, and it has its own variant. So it has got 3 variants. So again, it's a E484K is the deadly one. That's the one that makes it resistant. Just like the South African variant, this is also resistant to the AstraZeneca virus. By the way, the South Africans have stopped using AstraZeneca virus because it doesn't seem to work very well. Now, you should understand that variants can independently arise in different locations and need not necessarily be transported. Just because you find a South African variant in Toronto, it doesn't mean somebody from South Africa came here and gave it to somebody here. It can independently start in Toronto. So that one, right now they are finding out that there is no sort of background of people having this, having traveled anywhere. How did they get it? Because it independently arose in this area. Pfizer-BioNTech has just published a report that they modified the vaccine that works on the South African variant. That's the beauty of the whole thing, because all these virus vaccines have a whole package, a platform that they have developed. So all they have to do is change the one part, antigen part alone, and they can produce a new vaccine. And this new vaccine will work against the mutant. And they can use this as a booster shot. And they can protect the person. So it's a thing that we have to keep on track of that and keep, sequencing these things to find out what type of, stuff is seen in the public and what type of vaccines we have to use. So we can do all this. And just this morning, I was reading that the E484K, the South African mutation, which is the worrisome one, has been found in the UK variant as well, in the United Kingdom. So in other words, it's all over. So we have to sort of target this particular one, and we have to make this. And God knows what's going to happen the next time. They may have a newer one. So we have to be, alert to other changes of these mutants. And we have to, this kind of sequencing is very easy these days because they have machines to sequence this, and you just put it in. They have algorithms to read the stuff, and you can get it out in no time. Almost every lab has it. We have that in Sault Ste. Marie when we were doing the work to sequence genes, and it's very common. Okay, now I can't see this one. Oh, jeez. Many of the vaccines have been developed very, very fast because of the earlier research into developing many other vaccines. Stringent regulations of approval process provide safety. Most of them require two doses, but like the Johnson & Johnson vaccine, they require 1 dose. UK, South African, Brazil variants increase infectivity. Current vaccines are effective against the UK variant, but the South African and Brazilian variants are worrisome. These type of variants are to be expected. Tailor-made vaccines and cocktails as booster shots are probably the answer. Since the platforms are already there, modifying them can be done rapidly. Pfizer-BioNTech has just reported a new version of their vaccine that will work on the South African variant. There are new developments such as the antiviral PEG interferon lambda as a therapeutic to treat COVID patients. It's a nasal spray that blocks COVID-19 has been announced from the University of Birmingham research. If enough people are vaccinated, we might achieve herd immunity. That is here, these are all the people who have been vaccinated, and the guy in the middle has not been vaccinated, but he will be protected because there is no COVID-19 around the whole place. Now, there are a lot of people who are allergic and they cannot stand the vaccine, and they will all be protected because of herd immunity. So this is the goal we are looking forward to. Thank you. Are there any questions now?
Good job, Arthur.
Hi, Sadar.
Wow.
Where are you? I don't see you.
I have a question to Arthur. Can you repeat that, please?
Kudos. Yeah.
I'm sorry. I thought it's a sort of a complicated subject. I did the best I could, but you don't have to know all the details, but you just get an idea of what's happening.
That's why I went to debits and credits. I didn't have the brains for that. Yeah.
No.
Arthur, I have a question. If you could have any one of those vaccines, assuming they all cost the same, which one would you choose?
But they don't cost the same.
I was actually going to choose AstraZeneca, but now I have second thoughts because AstraZeneca doesn't work on that particular mutant that seems to be now in every virus, including the UK virus, as of this morning.
Yeah. So would you choose the Pfizer one, or
we have to use for the I guess the Moderna or BioNTech vaccine, Pfizer vaccine are probably the best bets. We do not know whether the Johnson & Johnson people have tested it against all these variants, but these things could change. The only thing is people will be constantly developing new vaccines, and we can always have a booster shot. And so we can get the vaccination that way. So the faster we do it, the better it will be. But unfortunately, Canada is behind the 8 ball.
Yeah. I think we're behind the 22nd ball, actually.
Yes.
Not the 8th one. But Arthur, a question on that. If we were to, with this process, this platform, this would effectively be like the flu vaccine. Every year,
yes,
you get a booster shot.
Exactly.
With whatever variant seems to be most prevalent.
Yes. Okay. We were naive to think that there won't be any mutants.
Yeah.
Initially there wasn't any, so they thought, oh, the spike protein is so specific, nothing can happen. So if we have, that is the Achilles heel. If we attack that, we will have the vaccine pat. But unfortunately, that's not what's happening.
Arthur, it looks like we're the guy in the middle here at the moment. So once the rest of the world gets vaccinated, because it doesn't look like we're going to get vaccinated anytime soon, we'll have the head thing all the way around us, right?
Well, the thing is, I think it all dates back to the time of Harper when he cut all this funding for research.
Yeah.
And when all the experts left the place. Now, we were the ones, the Winnipeg lab was the one where they developed the Ebola virus. And we have the expertise here in Canada. And that guy who was there moved to university, I think he's in Laval University now, because he thought, things are not, he wasn't getting any support. Now, that's a bad thing about us. The conservatives have a tendency to sort of cut, all this basic research. They say, why are we doing this hairy-fairy basic research stuff? Unfortunately, that's where all these vaccines come from. So now I think we have learned our lesson. I think next time around we shouldn't do that. We should have our own capacity. Now, we had Connaught lab there that they were producing vaccines. They have disappeared.
I think Frank has a question.
Yeah,
yes.
That was absolutely mind-boggling to see the advances that we've made in medical science. So we have to ask the question, if you wind the clock back to 50 years ago, should COVID have hit then without all this power that we had and all this computerization? It seems to me that could be the end of humanity.
No.
What would take place? You don't have that structure. I mean, what you've told us is absolutely mind-boggling. But we didn't have that 50 years ago.
But look at the standpoint from the virus. The virus, in order to exist, has to have a host. If it kills all the hosts, it will die. It will commit suicide. So the virus that evolves very well is the one that can coexist. So it'll give you a little bit of problems, but it'll keep the host alive so that it can survive too. Right. That would be the ultimate evolution of the virus.
Yeah, that would be the most.
It'll take a long time though.
Answer question.
Like malaria is a good example of a.
I think Gerd is talking, but he's not.
Answer the question.
Yes.
Arthur, if we are now starting to develop a vaccine in Montreal,
yes,
will be ready in 2022. We may be just in time to start an annual COVID vaccine. If ours is ready in 22, that's where we will need this new booster shot every year.
Well, there's a lot of unknowns here. We do not know how these booster shots will work. We do not even know how long these vaccines will last. Even the vaccines that they're giving right now, we do not know how long they will be effective. Although they think it will be effective for a long time, we are not sure. So boosters may be necessary, but there's a lot of unknown factors. But these vaccines that are being developed in Montreal, they are already in the system. It's only a question of the mechanical stuff that they have to put in place. So I think this will be faster than 22. I would say by the end of this year, we should be able to have these vaccines going.
It's the actual manufacturing that's taking the time to build the factories, right? So
yes.
Not the actual vaccine that's developed.
It's an old factory that has been closed down. It was an NRC lab. So thanks to Harper.
Gerd has a question.
Yes,
Gerd. No, it's not a question. It's really a comment. I want to go back to Frank. Yes, it's mind-boggling advances we have made in 50 years or longer. It's also mind-boggling advances we have made in all kind of other things. For example, with our data processing capabilities, with our communication, with all of these kind of things. It's mind-boggling. Yet, what is right now our only protection from this virus? A little piece of cloth in front of our face. I think that is so mind-boggling that we think we have so amazing progress made, but as Arthur said, first of all, we don't know what's going on at all, just a little bit, but we don't know the consequences. And what we do about this, we have to wear a little cloth in front of our face. Yeah. That is much more mind-boggling than anything else to me.
Something so simple, right?
Wash your hands too.
Yeah.
Danny had a question.
The question from Susan Marie, Ontario.
Okay. You're a tissue culturist, yes.
Arthur, first question is, you mentioned about choosing which vaccine you take.
Yeah.
But I think beggars don't have choices. Canada doesn't have many vaccines.
That's right. Yes.
So we just get what we get.
Yes. Yes, I think you're right. We have to take whatever we get and then wait for a booster shot. Glennie.
I have a question. Why were you partial toward the AstraZeneca?
What, beg your pardon?
Why were you favoring the AstraZeneca?
No, the reason I liked AstraZeneca was a little bit of a background to that. When the Oxford virus was developed, they had some kind of utopian standards. They said, this vaccine, we are not going to make it for profit. And we want companies to work with us only who are not involved in profiteering. So that's how they started it. And and it's going to be for the world. So they had very high moral standards, which appeal to me. And as far as I'm concerned, all these vaccines are going to work in various, extent. So if I had to choose, I would rather go for AstraZeneca.
Even with the, you wouldn't do that today, though, with the news.
Today, because of the South African mutant, it's creating a problem.
Right.
But, yeah, we will see. Every day you get new information. Like this morning, they said that the UK variant has the South African mutant.
So, Arthur, with all the variant, we have a new variant. We have the Brazilian, the UK, South African, and, one taking a vaccine today, and then they have a second dose to take. And if these variants are not part of that second dose, or they are, but a new variant come in, then we're going to have a booster. That's what you're saying.
That's what I think. That's what I think will happen. And I'm only, it's a hypothesis. I'm not sure what will happen, but the best thing that they could do is keep up with all the mutants as they come along. And since they have all the platforms ready, they can very quickly manufacture a new one. And since the safety of all the carriers, everything has been established, changing the antigen alone should not make any difference on the safety concerns. So they should pass through very quickly, and they will be able to get on top of it. And they can get a booster shot, and that will probably protect you.
So if you have to do it today, if you have to go out, not today, but go out in the next couple of weeks and get a vaccine, or next month, if the vaccine is available to you, what would you do as a scientist and a Rotarian? Which one
I would take whatever is available. I'll take it. But as Gerd says, I will wear that little piece of cloth on my face.
Arthur, did you read about the World Health Organization report the last couple of days saying that this virus did not come out of the lab in Wuhan?
Yes, I knew it long ago, because when you start fiddling around with the virus in the lab, there are always telltale signs. If you change a vector or do something, there will be little pieces of thing left. Just like when you launch a satellite, you have all these pieces floating around. There will be things floating around in the cell. And you can easily detect these little pieces here and there. And you can tell whether this virus is being manipulated. And in this Wuhan Virology Institute, they went and looked at it. And initially they all said, and I have a good friend in Sault Ste. Marie, Basil Arif, Sadar, our colleague, and he used to go to, Wuhan, and he was working at another virology institute there, and he was telling us this new virology institute is an international institute established by France, and they have people from all over the world working in this place, including Americans. But thanks to Trump, they were all pulled out of the place. So he sort of, he cut his own throat. And all these international scientists, if there's anything there available, hanky-panky going on, this should have come out a long time ago. You know, so there is no evidence for this.
What about there was a lady doctor? She said it did come out of there.
That's a French one, eh? Is that the one French one who said?
No, I think she's Chinese. I think she was Chinese.
Chinese, eh? No,
but Chinese name anyway. I don't know.
Yeah, I don't think there's any evidence. I was when I saw this, I said, yeah, this is what we all expected.
Yeah, Arthur, I have a question. This whole virus thing, this plan by the Illuminati that is trying to knock off half of the world's population, right? I mean, scientifically, it's a very good idea, isn't it? Back to Malthus.
Actually, you know, nature has a way of controlling population. Some of them are horrible. They have wars. When we study evolution, they always tell you, how do we control population? You have a disease, wars, and these are pandemics. This is how population control works. So in the grand scheme of things, Mother Nature is just playing its normal cards.
Okay. Now that we've got to the Illuminati question, I will ask Danny to thank the speaker.
Another question.
Well, Arthur, I got to tell you, that was a fantastic presentation. Although, in my simple mind, this is why I went into debits and credits. I was, thank you very much. Actually, you have done a marvelous job in terms of preparation and presenting the material in the most simplest term for most of us. I did got to say, some of it just went, but I do understand that, it's the kind of information that you have passed on. But with the questions and all of that, I did understand some of the ramifications of it. So I want to say on behalf of the club, thank you again, because I know you put in a lot of work in preparing this presentation, and we really, really appreciate it. Thank you very much.
Thank you.
Thank you.